Good morning, everyone, and welcome to our session called Accelerating Progress Along a Path to a Cure. My name is Ashley Cooney from the University of Iowa, and I will be co-chairing this session with Ross Wilson from the University of California, Berkeley. I'd like to first welcome you to this exciting session. I'm excited we have a lot of really invigorating talks to share with you today. The session features preclinical research focused on developing therapies for CF, and the topics include a super-exon approach, immune modulation to enhance secondary gene transfer, autologous stem cell gene therapy platforms, mRNA delivery, and delivery of suppressor tRNAs by picovectors. And I really want to highlight this work because it's all very important to advance the Path to a Cure mission to restore CFTR in all people with CF. We have a few educational objectives today to present new in vitro and in vivo models to test CFTR complementation. Second point is we're going to learn about immunosuppression for repeat delivery and discover RNA-based approaches to restore CFTR. We're going to discuss gene delivery or repair options for the final 10% of people with CF who do not currently benefit from modulator treatment. I'd like to also point out that we will be taking questions after each presentation, and we're going to be doing so through the center microphone in the room or through the NACFC app. We'll be monitoring both. So the instructions to ask through the app: open your mobile phone, locate session W29, and click the Q&A button on the navigator bar, and we'll be able to see those questions up here. This is our list of presenters, and we first have Craig Hodges from Case Western Reserve University with his title as a CFTR super-exon strategy in W1282 intestinal organoids and mice. So Craig, if you'll come on up.
All right. Thank you, Ashley. So first, I wanted to thank the moderators for allowing me to speak about our work today, really the super-exon work in the W1282X format here. Really, this is a joint effort between the CFF-T lab, those names in gray, as well as Case Western Reserve, the names in blue. This came about because some findings that the CFF-T lab had very interesting findings with the super-exon in vitro, and they wanted to translate that in vivo, so had talked to us about mouse work. So I have no disclosures. So you've probably already seen this in one form or another, but essentially right now we're at about a little over 90% of CF patients are eligible for FDA-approved modulator due to the mutation that they carry. That's terrific, but as the CF Foundation has stated, we're not going to stop there until 100% of patients have a cure. So right now there's at least 5%, depending on the estimate, maybe around 10% of patients that are not eligible for modulators due to the mutations that they carry. So how do we restore CFTR in the remaining patients? It's not an easy answer, mainly due to this slide. It's a complex problem because there are so many CFTR mutations throughout the gene, as shown here, the gene at the top, and then all the dashes at the bottom are, or below it, are all the different mutations that have been identified in CFTR. So there's over 2,000 mutations encompassing missense, nonsense, frameshift, splicing mutations. Of these 2,000, at least 340 mutations have been shown to definitively cause CF. And of those 340, a little over 50% actually have modulators that can be used on them. So that leaves a lot of mutations that fall within that last 10% of patients that we have to figure out how to restore CFTR function. So there's a lot of strategies. Only a subset of them are listed here. So for instance, for nonsense mutations, you could use a translational read-through strategy, NMD, or nonsense-mediated decay inhibition, as well as a tRNA strategy to suppress nonsense mutations. You could use antisense oligos. We've seen some talks already about using that for splicing mutations that seems to be quite successful. as well as you could go and gene edit all of these mutations one at a time using either traditional CRISPR-Cas or base editing or another gene editing tool. However, I think what you saw in the plenary yesterday that Dr. McRae essentially introduced this topic and said, what about a mutation agnostic approach where one strategy can restore CFTR function to many or most CFTR mutations? And so the strategy he mentioned, or one of the strategies, was a super-exon strategy. And essentially what this is an approach that circumvents the endogenous mutation by inserting a cDNA somewhere before the mutation in the gene. In this example, we're inserting exon 23 through 27 in a super-exon before the W1282X in exon 23. Because this insertion includes a splice site and a poly-A signal sequence, the hope is that once inserted, that the final mRNA spliced product will include the endogenous exons 1 through 22, as well as the super-exons, the exons in the super-exon 23 through 27. So as this is written, this strategy would help about 15% of the CFTR mutations out there. If you did the same strategy right after exon 1 and used a super-exon for 2 through 27, that would be close to 99% of the CFTR mutations. So this is a strategy that has been used in at least 3 different groups. Some of the people who led this work are in this room, so I expect some questions. But these 3 papers that have come out all used a super-exon strategy. They all used a different super-exon with different mutations as well as cell types. So the first group at the top used a super-exon 11 through 27 and a CFBE cell line with delta F. The next group used an exon, super-exon containing 9 through 27 exons in the delta F primary airway basal cells. And then the last group, which is the most recent paper that has come out, they used a super-exon containing 23 through 27, and it was in a 16 HBE gene edited cell line that contained W1282X. So all of these used a different strategy, sometimes even a different insertion strategy. The components of the super-exon were a little bit different, so it's kind of hard to compare, but all of them showed successful CFTR restoration ranging from 10% of wild type to very similar to wild type levels. And so at the CFFT lab, what they suggested, or what they wanted to show, was really to develop and optimize a blueprint for an effective super-exon, because we really don't know the best components that should go into the super-exon. And then they wanted to share this blueprint with other groups, especially those groups that are involved in delivery. And the way that they did this was they used 16 HBE cell lines that were gene edited to either have W1282X or N1303K. And so these are 3 of the super-exons that they were focused on. Each of them was a little bit different in the fact that either they had a native splice site or a synthetic strong splice site shown here, and they differed by either having a bovine growth hormone poly-A signal sequence or the SV40. Both of those are commonly used in transgenes. And then they would be inserted in intron 22 before either a N1303K mutation or W1282X. And if everything worked as we expected, then you would get a full-length wild-type CFTR. But if the poly-A or native splice site didn't work quite as well as we expected, then the final mRNA would include either the N1303K or the W1282X, depending on the cell line that you're using. And so here is an example of the functional analysis that they did using a transepithelial electrical conductance. This is a wild type 16 HBE cell cells where they added forskolin, and I'm sure you've seen this before, but where you add forskolin, the current goes up. You add VX770 and goes up even more, and then you can inhibit CFTR with INH172. Now, if you have a 16-HBE with the N1303K and you do these same manipulations, you see no increase in current, suggesting no CFTR function. But what they did was they inserted this super-exon and identified 3 independent clones, shown here as 3A, B, and C, and then looked at CFTR function. And what they saw was that super-exon restored function from anywhere from 50 to 100% wild-type function in these in this cell line. They are still trying to figure out why the variability is there with the different clones, so we don't know the reason for that, but it's very promising for as much CFTR function that was restored. So I am summarizing a lot of their work, so please don't come up and hit me afterwards, Hillary, but she is giving a talk later on Saturday, so you can look at some of this as well. But essentially the 3 super exons that they tried, the littlest, the one that restored the littlest amount of function was the one with a native splice site and a bovine growth hormone poly-A, whereas the one that had the strongest, surprisingly, was the strong splice site and maybe the surprising part is the SV40 poly-A for those of you who have used that. And so this is when we got involved and the CFFT lab came and said, you know, we'd really like to translate this in vivo. What happens if we take this same super-exon that they saw a very strong functional level with and put that in a CF mice? Would we get CFTR restored? What would the manifestations look like? And so we wanted to do this in the context of a W1282X mutation. We've actually made this mouse model and just with the W1282X. And what we've shown is that it has little to no CFTR function. These are intestinal sections where you look at change in short circuit current after forskolin to see if you have any CFTR function. And compared to wild type, W1282X really does not. Growth is reduced just like you would normally see in a CF mouse, as well as intestinal obstruction occurs, so survival is much reduced. And so I have question marks on all of these because we don't know if you have a super-exon in this context, what it would look like. Would all these manifestations go away? And that's the question we set out to answer. So Ron Conlin and myself came up with a transgene to insert into the mouse to hopefully answer this question. So this shows a targeting vector where on both sides there are arms that have homologous regions, similar or homologous regions to the mouse chromosome. And then we used a intron 22 guide RNA as well as Cas9 and the super-exon targeting vector to insert this whole vector into, or the super-exon into the mouse genome. Now we did this in one-cell embryo. So we kind of cheated for all those delivery folks. We just injected it right into the one-cell embryo. And I realize that's not how it's going to take place in CF patients. But this isn't a delivery talk. This is more of a, will the super-exon work when you're given the best strategy out there where you have the super-exon in every single cell. And so in this is on a wild type mouse CFTR, so there's no CFTR mutation yet. We wanted to see if human CFTR was expressed. And what we found was in these mice that had one copy of the super-exon and the other copy had a wild type allele, so 2 alleles. And we used digital PCR to try to estimate how much was coming off this super-exon allele. Compared to totally wild type mice, what we found was that, at least in the ileum and the lung, we had about 35 to 40% of CFTR function as a percentage of 100% wild type function. So you might expect closer to 50. So certainly there could be some missplicing occurring here, and we're going to look more into that. Look at further tissues as well as homozygous this mouse. So both copies have the super-exon allele, and hopefully we'll have a better idea of what's going on there. The next part of this was to insert the CFTR mutation back into this mouse. So we used a whole bunch of embryos from containing the human super-exon 23 through 27. And then we introduced a guide RNA, the W1282X mutation on a single-stranded oligo, and essentially got this mouse. Now, many of you might have said, why didn't you just do that in the first place? Well, we tried, and we just didn't get the right mouse. So we had to do it again a double insertion, essentially. A little bit harder, takes more time, but that's the way it goes. But right now we do have actually quite a few founder mice with this super-exon and CFTR mutations. We're crossing that out, and hopefully we'll have an answer in the next 4 to 5 months. Maybe I can put that in the next abstract that I'm sure will be due on Monday when we get home from this meeting. So in the meantime, while we were making this mouse, we also wanted to look at primary cells. And so these were the 3 exons again, from weakest to strongest based on the CFFT labs results. But we also wanted to do this with mouse exon 23 through 27 as a positive control, because we didn't want to see, let's say, lower function and blame it on, it could be blamed on a chimeric protein between human and mouse. So we used all 6 of these exons in intestinal organoids. So for those of you who are not familiar with this, these come from stem cells in the intestine. We use the small intestine in the mouse, and they really, in the right conditions, if these intestinal stem cells are grown in the right conditions, they'll form intestinal organoids. And these have crypt-like structures, an internal lumen that with differentiated cells, and really a CFTR is predominantly expressed here in the apical membrane of the crypt cells. And when you activate CFTR, you go from an organoid that looks like this to an organoid that is swollen, and that's due to a CFTR driving secretion of electrolytes and fluids. And so this is called enforskellin-induced swelling assay. And in a wild type mouse, what you'll see is a nice swelling like this, that movie on your left. That's what happens. CFTR works just fine, but in and it swells within an hour. With a W29 mouse, however, that is actually going forward, but you don't see anything because it's a really boring movie. But it is actually, forskolin is on there. And so when you look at this as a percent area increase, wild type goes up, W12A2X does not. The great thing about organoids also is that they're pretty easy to gene edit, at least for us. And so you take these organoids from a W12A2X mouse, you put them as single cells, you digest them, and then you go ahead and use a guide to intron 22, a Cas9, and either a human or a mouse super-exon with electroporation. And the great part about this is you can plate all these cells out, and every single cell that's a stem cell will grow into a whole organoid. Then you add forskolin, and anything that has CFTR function will swell, and anything that doesn't. So then you can just take your pipette, pick the swelled organoids, subclone them out to a whole population that has the same genetics, and then you can do another forskolin-induced swelling and sequence. And so what we found with this first super-exon that had the mouse 23 through 27, what you'll see is that it swelled just fine. And compared to wild type, it was very similar. So it did restore CFTR function in these organoids. And in the next construct was the human 23 through 27, the same one that we inserted in the mouse, and we see plenty of function. So we were pretty happy with that. And this here are the triangles, very similar to wild type. Now, what I will say is that this is the area under the curve. If you were to quantitate these, this is wild type W12A2X, and this is over an hour long. Really the human super-exon, mouse super-exon, and then we added on an edited W12A2X mouse that we used traditional CRISPR-Cas to change it into wild type. They were all very similar. Now we were using a higher dose of forskolin, so we are going back and using a dilution curve to see if there are subtle differences in this assay. So I've shown you those two. The other 4, we've actually got swelling as well. We just haven't done it enough time for me to show you that data, but they do swell the W12A2X, and it does look like, just based by eye so far, that they may not be as strong as these two. So in summary, I've shown you that a super-exon strategy can be used to restore CFTR function in the rare alleles that are ineligible for modulators. Super-exon components do matter. So Hillary and her team really showed that the splice site and poly A does have a large effect on restoring CFTR function. And please attend her talk on S25 on Saturday morning, because she's going to tell you a lot more about what I presented. She has a lot more work. That group does. And then, can we restore CFTR function in vivo? I'll tell you about that next meeting, evidently. I just want to thank all the people on this slide, especially Hillary and Cal and Ron Conlon, but really all the people are indispensable on this slide, as well as the funding. And then I just want to comment that if you need CF mice, I do run a CF Mouse Resource Center funded by the CF Foundation, so you can contact me or go to our website. So otherwise, I'll take questions. Thanks. And I know you all felt cheated because you thought you were going to come here and see the answer to, if it restored it in vivo. I wish I had that information. Sorry.
Hi, Margarida Amaral from Portugal. Very nice talk. I was wondering what's the forskolin concentration that you use on the organoids and whether you see for the wild type ones pre-swelling.
Yeah, so for mouse, we actually have to use a higher concentration than human organoids. I know they use a lot less. We use 10 micromolar for mouse.
Wow.
Yeah, it's like blowing it out, but we can go all the way down to, we've gone as low as a couple nanomolar, and you can get swelling. And we do not see the pre-swelling in mouse wild type in the conditions that we use. We have tried a couple different conditions and seen pre-swelling like the human organoids, but for the most part, we keep it in the conditions where there's no luminal swelling before. And we realize 10 is like turning on a fire hose. So we're going to try to use a garden hose next.
Thank you.
Hey, Craig, really great talk. And so really nice proof of concept data. As a delivery person,
I cheated, sorry.
No, that's okay. What size cargo are we talking about in order to deliver this?
So as far as the base pairs involved, or?
Yeah, the base pairs are like this, the size of the construct that you'd need to deliver as a therapeutic.
So for the exon 23 through 27, trying to think so we had the splice site and.
Okay.
All right. I was about to say, I was trying to do it. So 1.5, so, but it would be even bigger for a little larger exon that you have, the 2 through 27, right? So much larger. So 1.5 for the smaller one that I've introduced here. But
Okay, perfect. Thanks.
Sure. Hillary couldn't stand seeing me trying to do the math. Sorry, Hillary. No, it's okay. Thank you for the save.
Fantastic presentation, Craig. As an ion transport person, I can't help but wonder, and this is a little bit off topic, whether it's actually bad if you have too much CFTR and whether there might not be an internal mechanism that tries to keep the function down. I mean that's not the purpose of your project.
Right. We're actually working on another project with the CF Foundation where that is the exact question. Is having too much CFTR a bad thing? And so we are making a mouse that overexpresses human CFTR. And maybe I'll put that in as an abstract next year. I don't know.
Awesome. Thank you.
Yeah. So hopefully we can answer that question.
Yeah, nice work. Since you're directly injecting the DNA right into the cells, you have a nice opportunity to find out about off-target effects or if there's fragments of the super-exon that's incorporated and so forth. I know you didn't maybe do too many cells because you're trying to get a live mouse, but had you found any evidence that there's some problems when you do this, or did you sequence the entire genome?
It's a good question. I always say for off-target effects, I don't care so much in the mouse because I'll just breed them out if there's any off-target.
We kind of care in humans, so.
I know. I cheat a lot. But we haven't looked into that. We do see every once in a while a randomly integrated super-exon, but haven't seen any issues with that yet.
Yeah, particularly if it stays around in the cell, it's going to have more and more opportunities, right, for integration over some days.
Exactly.
So
exactly. And
got a good system to test that.
Yeah, we haven't looked at that particularly, but it's good to bring up.
Great. Thanks, Gary.
All right, I think we have time for one more question.
Sorry. Not a question, but just a technical comment. This is down the line, but we've noticed that not all poly-A's are captured as efficiently in single-cell sequencing, particularly the rapid globin. Poly-A isn't captured efficiently at all. I don't know about growth hormone, but if we envision doing single-cell in clinical trials down the line, it may be good to know whether how efficiently the poly-A tail is captured, and that's
a really good point. Thanks for bringing that up. Yeah. So. All right. Thank you very much.
All right. Next up, we have Robert Clark, who is at Tulane in the lab of Jay Coles, and we're going to hear about some strategies to potentially allow redosing in the lung. So, yeah, take it away.
Yeah, thank you. Let's see, how do I? Where do I? Hello, everybody. Thank you for being here. And I'd like to thank the moderators for this opportunity to present our work. I'm an MD PhD student in the lab of Dr. Jay Coles at Tulane, as you just heard. And I'm here to talk about our work with using anti-CD20 to try to re-administer gene therapy vectors. I have nothing to disclose. So as you've all heard probably many times now, 8 to 10% of people with CF have undruggable mutations, and not everybody responds to correctors. And inhaled gene therapy can present a genotype agnostic method to correct some of these mutations. However, one of the most commonly used tools, AAV, in other trials, host immune responses have been indicated as a really primary barrier to effective and durable treatment. So we thought that perhaps some of these issues could be overcome by redosing. One paradigm that emerged from some of the hemophilia A trials is that if you give really low doses of vector, you get insufficient correction and no therapeutic efficacy. And high doses result in the stimulation of immune responses, which eliminate the transduced cells. And again, failure of therapeutic efficacy. So redosing, one thing it would do would be to permit low and slow dosing strategies so you could reach the minimum effective dose without triggering these immune responses. Another concern with the lung is that treatment may wear off over time, and so you might have to re-administer several times, like over the course of a lifetime of treatment. And one thing is all of the immune responses to AAV largely have been studied in the systemic context with delivery to the liver, but we are concerned that there may be more robust immune responses that are present in the lung. And this is obviously a particular concern with the inflamed environment of the CF lung. So just a primer on immune responses in the lung. So in the lung, local antigen encounter will recruit circulating antigen-specific cells and elicit these T and B resident memory cells, which reside in the lung parenchyma ready to respond rapidly upon antigen re-exposure. These, we believe, may present a barrier to effective gene therapy. And one kind of technical note here is we use flow cytometry extensively to identify these cells. And one of the main markers we use is CD69, which kind of marks all resident memory cells in the lung. And then T cells can also be marked by CD103. And also to identify this, we inject mice prior to euthanasia with an anti-CD45 antibody to label all circulating blood cells to exclude them from analysis. Another unique thing about the lung is that B cells can play a key role as antigen-presenting cells, and Dr. Coles' previous work has shown that this is particularly important for fungal pathogens such as Pneumocystis marina, but it can also happen for viral-like particles as well. So we therefore sought to understand whether anti-CD20, which clinically is approved as rituximab and has been used safely in people with CF, would be able to block both B cell and T cell mediated responses to gene therapy vectors. So here's our experimental design. Basically 2 days before dosing with a first-round adenovirus, we studied adenovirus first because the immune responses are kind of more robust and well-described. And we thought that if we could block those immune responses, we would be able to likely block AAV responses as well. So yeah, 2 days before dosing with the adenovirus, we gave anti-CD20 a high or low dose or an isotype control. Over the course of the experiment, I sampled the peripheral blood to assess B-cell depletion. And then at euthanasia, we had several outcomes. We did flow cytometry on the lung and bronchoalveolar lavage to identify the lung-resident lymphocytes. And we performed ELISA on the serum and bronchoalveolar lavage to look at the anti-adenovirus immunoglobulin responses. Also, we did single-cell RNA sequencing of some of the parenchymal lung T cells to see whether we were really eliciting T resident memory cells. So first, this was just kind of validation, looking at the blood counts of CD19 cells over the study period. We found that the high dose of anti-CD20 was able to suppress the peripheral circulating B cells for the entire month-long period. But the lower dose, we started to see some recovery at the end. Also, we did not really see a systemic T cell proliferative response in response to adenovirus delivery. So we first looked at the humoral response, the B-cell mediated response, which we'd expect to be reduced based on giving anti-CD20. And that's exactly what we saw. We saw a pretty good reduction in serum Ig responses, though not an entirely complete reduction. It was about 80%. But we did see a more dramatic reduction in the airways. The IgG and IgA in the bronchoalveolar lavage was pretty much completely wiped out. We also looked at the B resident memory cells in the lung and the BAL, which again we defined as these class-switched CD19 positive cells, which excluded the labeled circulating blood cells, and CD69 positive. And in both cases, we see that adenovirus delivery will elicit an increase in the number of lung resident B cells in both the lung and the BAL, and that's ameliorated by anti-CD20 treatment. And then this was the single-cell sequencing data. We compared the single-cell sequencing of the lung T cells to naive spleen, and we found that these lung T cells were much more oligoclonal. There were some highly expanded clones, and we want to look at those in more detail because we figured those highly expanded clones would be the antigen-specific cells that we were interested in. We saw that most of them fell within the CD8 population, which is what we'd expect to being a viral infection. And then we simultaneously looked at the transcriptional profile of these cells, and as you can see, they cluster very differently than the cells from a naive spleen. And we examined this cluster for the canonical T-resident memory cell markers. There's CD69, CD103, and CD49A, which were all upregulated, and then these are two genes that are known to be downregulated in T-resident memory cells, and similarly, those were downregulated. So we feel confident in calling that a kind of a T-resident memory cell cluster. We also looked at the impact of anti-CD20 treatment on these T-resident memory cells, on the CD8 population specifically. And one thing we did note is that we see this decrease, and these are the CD69, CD103 double positive cells, which are thought to be kind of the longest lived and most ready to rapidly respond T-resident memory cells. It didn't bring it quite all the way back down to the levels that you find in naive mice, but it did reduce it. And then in the bronchoalveolar lavage, that was also reflected. So then we sought to understand whether anti-CD20 could rescue second-round gene transfer. So similar to before, 2 days prior, we dosed them with anti-CD20 or isotype control. And then a month later, instead of euthanizing them, we dosed them with a second round of vector expressing adenovirus LAC-Z. And we looked at IHC for transgene delivery distribution, and also did an activity assay on the lung homogenate to see kind of what level of second round gene transfer we were getting. So we can see that we're hitting the airway cells that are relevant for CF. So that's like the right place. We're happy to see that. And then we actually were able to achieve pretty close to complete rescue of second round gene transfer, which we were very pleased to see. So we wanted to kind of understand why this was. And since we saw the biggest effects in the humoral compartment, we used a series of mouse lines which have mutations in immunoglobulin-related genes. So IgA mice lack serum and secreted IgA, and then FcRn mice lack receptors which transport IgG across epithelial barriers, including in the lung. And then AID-MUS mice lack all secreted immunoglobulins. And so we did the same redelivery assay, and we found that only the AID-MUS mice were able to be completely redelivered to. So this points to a critical role for secreted immunoglobulins, but also maybe says that in the absence of IgA, IgG can compensate, and in the absence of IgG, IgA can compensate. So from our adenovirus experiments, our kind of takeaways were that the luminal immunoglobulins may be more important than systemic immunoglobulins in preventing redelivery. Because if you remember, we only saw that like 80% reduction in systemic immunoglobulins, but we saw a complete rescue of redelivery. And we saw complete abrogation of the luminal IG responses. Anti-CD20 also prevented the formation of anti-vector B-resident memory cells. And then the impacts on T-cell mediated immunity was a little less certain. We were seeing some positive hints, but nothing quite so dramatic as in the B-cell compartment as we'd expect. So we next sought to expand our results to AAV, and this was kind of validation for serotype selection. We chose 3 different AAV6-based serotypes, dosed them with AAV, and this time we euthanized after a month because the kinetics of AAV expression are a lot slower than adenovirus. So adenovirus, you can get really high signal after 3 days, but you need to wait like two weeks to a month for AAV. And based on this, we moved ahead with using AAV 6.2. So we carried out the redelivery experiment, and this time we again dosed 2 days prior to vector delivery, but we also dosed 2 days prior to the second vector delivery because we wanted to make sure that these mice stayed suppressed the entire study period, and we had to have this longer 3-week period to analyze transgene expression. We also switched to BALB/C mice for these experiments because they have a mapped CD8 cytotoxic T lymphocyte epitope for AAV, so we'd be able to assay antigen-specific responses, T-cell responses in greater detail. So similar outcomes as before with flow cytometry, and then also for antigen specificity, ELISpot did a luciferase assay and the ELISAs. So again, the B-cell humoral response was kind of as we'd expect. There were a couple differences though versus adenovirus. So again, anti-CD20 depresses the number of BRM cells in the lung. But when we looked at the ELISA data, so after only a single dose of AAV, the serum IG, like the IG responses in all compartments are not nearly as robust as adenovirus. We do see a robust increase after 2 doses, which is ameliorated by anti-CD20, but also particularly in the luminal compartment, the single dose doesn't seem to be strongly immunogenic in this model. We also looked at the CD8 T-cell responses to AAV. Similarly, we see an increase in the number of both total and antigen-specific T-resident memory cells after 2 doses of AAV. And we do see a slight reduction with anti-CD20 treatment, but it didn't really quite reach statistical significance. It was like 0.08. But then the next thing we did was look at the transgene expression. And here we got some results that we were not expecting. As you can see here, the isotype in anti-CD20 treated mice reach only about 50% of the transgene expression relative to the single vector only. This is definitely something we're exploring. It's interesting because all of the previous data in our thought is that maybe this is CD8 T cell mediated because we're looking at a period 3 weeks after the second round gene transfer. But all of the data in liver with AAV immunology that we have has kind of pointed towards CD8 T cells in mice being ineffective at eliminating AAV transduced cells. But this is in the lung, and obviously we're going to have different considerations, and we're really interested to kind of continue to tease out those differences. So conclusions from the AAV experiment, redosing of AAV 6.2 to the lung results in approximately 50% reduction in transgene expression. The mucosal antibody responses appeared to be less robust than the serum antibody responses. AAV 6.2 is definitely less immunogenic than adenovirus, and we believe that factors other than humoral immunity may limit redelivery of AAV 6.2. And in the future, we're also going to, I think maybe some of you heard from Dr. Coles yesterday about the eyeball structures, which are kind of, they're like Peyer's patches in the lung, localized sites of antigen presentation. And they may, they're present ubiquitously in the CF lung, and they may present a large challenge to especially immunomodulation strategies like this. So future experiments, we're going to look and see if those structures will promote greater anti-vector immune responses. With that, I would just like to acknowledge everybody at the Center for Translational Research and Infection Immunity, and all of my lab members and everybody who's helped, and the funding sources and the Cystic Fibrosis Foundation. So yeah, thank you. With that, I'd like to ask any questions or take any questions.
Do we have any questions for our speaker?
There's one online that we can get started with. So someone asked, have you investigated whether the anti-CD20 approach will impact vaccine titers and whether patients will require redose of vaccines following treatment?
Yeah, so that's actually something that's been looked at in the context of COVID. And so definitely anti-CD20 affects antibody responses to vaccines dramatically. That was one concern with the COVID vaccines as well, because but people have done a lot of optimization with that. Some people want to pull people off their anti-CD20 treatment before vaccinating them, but you still get robust T cell immune responses, like for the SARS-CoV-2. But certain vaccines that are not gonna make intracellular protein are probably it would be worse. But also, like, it doesn't knock out pre-existing plasma cells. It only knocks out the cells that are that have not experienced antigen. So you'll still have all the pre-existing immunity from your previous vaccines. It's not going to affect that.
Okay, thanks.
I have a question if there are no other questions on the floor. Do you have another antibody that you're interested in looking at for AAV? Is there another CD20 version that you're interested in?
Yeah, Mina Hanna up there, he's a resident in our lab. He's also looking at IL-21 blocking antibodies. That one less studied, so it's definitely like more investigational, and we're really interested to see we expect that one maybe will have like more broad impacts rather than being solely in the B cell compartment. There might be more pleiotropic effects on T cells and other cells.
The FDA last week just approved anti-CD3 gamma for type 1 diabetes. So that may be another interesting molecule.
Yeah.
And I have one more question for you as well.
Yeah.
So the goal of suppressing the immune responses to get longer term expression, and adenovirus notoriously the expression will go away over time.
Right.
Did you look at how long the expression lasted after the second dose?
No, we haven't looked at that. We were kind of primarily focused on the proof of concept for adenovirus, and then going to move to AAV, because we recognize this is a problem with adenovirus and think that, probably that won't be the one that we'll move forward with in the future. So, but no, it's a great point.
All right. Thank you very much. If there are no other questions, we'll welcome our next speaker.
Thank you.
Oh, sure. Okay, so today we have Dawn Bravo from the Stanford University School of Medicine, and her talk is entitled Preclinical Development of an Autologous Stem Cell Gene Therapy Platform for Treatment of Cystic Fibrosis Sinus and Upper Airway Disease.
Thanks so much for giving me the opportunity to discuss our work on the development of an autologous stem cell gene therapy to treat cystic fibrosis, first in the upper airways. I have nothing to disclose. We have a great collaborative team at Stanford. Our gene therapy group, Matt Porteous, and now Assistant Professor Sri Ram Vedanathan, Tushar Desai, our pulmonology expert, our CF team, Carlos Mila, Jeff Wine, Zach Sellers, and Jeanette Baker from Bone Marrow Transplant at Stanford in Rob Nagrin's lab, and myself and Jekar Naik in the rhinology division, where Jekar performs endoscopic sinus surgery on the CF patients. So CF is a progressive lung disease as well as a sinus disease. And it affects hundreds of thousands of patients, but virtually 100% of these patients also have CF sinus disease that often require surgical intervention throughout their life. So Trikafta is a great therapy. However, it can't treat all patients, and this is due to those with nonsense mutations, as well as an intolerance to the therapy, as well as the expense of the therapy. So stem cell therapy such as this would be very attractive for these patients, and attempts to do in vivo gene targeting have had numerous complications due to targeting and immune developed toxicity. So we're proposing an ex vivo to in vivo stem cell based approach to treat first the chronic sinusitis in the upper airways, and we'll do this by CRISPR genome editing, which offers a trackable approach and a durable approach. And these stem cells, once embedded, should produce viable differentiated stem cells with restored CF activity and hopefully improved quality of life. Even though it's the upper airways, it might prevent lower airway infection. And because we're using the patient's own stem cells, there's little risk or low risk of immune response or rejection. And once we show that we're able to do this in the upper airways, we'll then have some information to more effectively target the more risky lower airways. So our overall plan then was to isolate a patient's stem cells from the upper airways, effective CFTR stem cells, correct them, and then transplant them back into the patient in an autologous fashion. And we'll first prove this in rodent models. So our overall plan is to isolate the upper airway stem cells, expand them, perform CRISPR gene editing, expand these edited stem cells, and then how do we transplant these into a rodent model system, finding the optimal scaffold reagents to do this. So Sriram Vedanathan was able to show in cell stem cell, he was able to correct the most common delta F508 mutation, and with great efficiency. He then, because we had so many numerous patients with different mutations, we thought, let's try to correct the entire CFTR gene. And Sriram was able to show this in molecular therapy recently. Sriram also engineered a truncated CD19 tag, which would allow us to track these cells and actually enrich for these cells. It's a extracellular tag, non-toxic. So he was able to get upwards of 60 to 80% enrichment for these corrected upper airway stem cells in both upper and lower airways. And Zach Sellers demonstrated that they had functional CFTR activity above, way above threshold level. So now we have our bona fide CFTR full-length therapy to go forward. So now we, after isolating the patient's upper airway stem cells, correcting the CFTR gene and adding different genetic tracers such as luciferase, GFP, and now a truncated CD19, we can expand these, explore the best biomaterials for scaffolds, and then transplant them into rodent model systems. So if once successful, then we will proceed to the upper airways, which is a non-critical organ, easy to monitor, easy to access, before we try this in the lower airways. So little concern for immune reactions because it's the patient's stem cells. We're targeting the stem cells, and we can characterize the CFTR efficiency, and now can we then transplant these stem cells? So first off, we started with okay, let's do a mouse-to-mouse model system first for proof of concept. And Jeanette Baker had engineered these GFP luciferase transgenic mice. So we thought we would use those. We could transplant them and then do live animal imaging and monitor the signal with luciferase. Also, we could monitor the GFP to see whether or not these cells engrafted correctly. Initially then we needed to condition the epithelium and to allow for engraftment. So we knew that SO2 gas was an efficient method to denude the epithelium, and also tried nasal brush, and both were very effective. As you see, very stable engraftment for 2 months, 3 months, and then up to 9 months. This lifespan of the animals, basically very stable. And we see that these stem cells, these GFP, now labeled GFP luciferase stem cells, we weren't sure where they engrafted to, but after harvesting, sacrificing the mice, we did in fact see that they embedded into the stem cell layer that is keratin 5 positive, shown in white, and all the progeny would then have GFP. In here we show streaks of GFP with alpha tubulin for ciliated cells. This is the black is the airway. And we also notice that these stem cells seem to, once engraft, stay in place because we see this very stable signal. They seem to, as stem cells do, just replenish rather than migrate and extend, and also we didn't see tumor formation. So we then wanted a more reliable method of injuring the upper airways that was clinically relevant. So we looked at polydocanol, and this would prove to be a very effective method. Again, we see very stable engraftment over time, up to 9 months. And also we can see by stereofluorescent microscopy the GFP signal in the left side of the nasal cavity where we engrafted these stem cells. And again, very stable over time. And when we open the nasal cavity through the midway section of the nasal cavity and open the cavity in half, we can see where these cells embedded. So the murine upper airways are composed of olfactory epithelium, squamous epithelium, and then in blue, respiratory epithelium, which is where we isolated our stem cells from. And we see they located to the respiratory epithelium in the nasal cavity. These cells embedded in various areas of the nasal cavity. This black space is the airway, and we see that the cells embed into the ventral walls, the septal walls, the two turbinates, and the lateral walls. And again, they give rise to the differentiated progeny shown with GFP, and in this example with alpha tubulin shown in red above the stem cell layer, the basal stem cell layer, keratin 5, and also give rise to goblet cells. So we have a proof of concept then, and we can proceed. We also demonstrated that this was a very efficient method over several experiments. And again, in closer look, we can see again that the basal stem cells shown in magenta are GFP positive and give rise to the goblet cells here shown in red, and the ciliated stem cells, or ciliated cells, in green with that barrier. And then this is just a more close view showing the ciliated cells and the goblet cells. So now we're ready then to proceed, now that we have proved that we can do this in a murine-to-murine system. We wanted to proceed to our human-to-mouse system. So we investigated several hydrogels as FDA-approved matrices to embed the stem cells. Fibrinogen and BioSilk were the most similar to Major-Gel, which is what I had used up until this point. And they both expressed our stem cell markers for keratin W29 and P63. Shriram tested various combinations of fibrinogen and thrombin, which is a surgical use, often used in the OR, and found the most optimal combination. And so from then on, we used the fibrogen thrombin method, as it was very similar to Matrigel. And by immunohistochemistry staining, we see that both embed almost the same. So now we have our embedding scaffold. Now we can correct our human stem cells, and Sriram Vaidyanathan also engineered a truncated nanoleukin GFP, so we could follow these stem cells within the hemoglobin locus. He also, as I mentioned, did these truncated CD19 within the exon 1, where he homologously combined two halves of the CFTR gene into exon 1. This would correct all the mutations, at least 99%. So we have our system ready to go, and it also is very functional with CFTR function. And so this is our, we're able to visualize with the truncated luciferase and GFP that in fact these human stem cells are giving us a signal for up to about 4 months, not as strong as mouse to mouse, but we might expect that with the non-species to species interaction. And so now we're ready to proceed then with our final product. This would be used in human. The truncated CD19 is non-toxic. It's extracellular. It's not going to cause signaling within the cells. However, it was very difficult for me to actually find this engrafted human stem cells because the CFTR signal was not that strong. So I had to use species-specific markers such as EpCAM, which stains the epithelium, compared with human-specific EpCAM. So I saw this island of what appeared to be human stem cells or human cells within the murine epithelium. These also stained for the CD19, and also they were in between the EpCAM murine signal, and also expressed MUC5AC in green, as well as ciliated cells. So these are serial sections. And then taking a closer look, we could see that actually mouse nuclei contain condensed chromatin, whereas human nuclei do not. So this was another verification that these are in fact human-derived differentiated cells in the mouse. We also stained for a human-specific nuclei marker KU80, and also we see a wide expression of MUC5AC for goblet cells. And these so far I'm showing are CF donors, but we get the same expression pattern in non-CF samples. So this is the CD19 tag, and this is the differentiated cells. Goblet cells in green, alpha-tubulin ciliated cells in red. So, and this is just demonstrating the engraftment. They engrafted as they did in the mouse, not to the highest degree that we saw in a murine-to-murine sample. But also we noticed that the goblet cells were about 25 times larger than the murine goblet cells. So that might be a reason that we don't get good engraftment with the cells. So we've reached a number of our milestones, and we've had our Interact meeting with the FDA, and they are very excited about our study and gave us the go-ahead to now do GMP scale-up and do our toxicity studies before the IND approval to the FDA to go forward. So with that, I'd like to thank our funding sources, our incredible CF team, and our current lab members and past lab members. And with that, I'll take questions. Thank you.
Jay Coles, Tulane. I just have a technical question. How do you brush the mouse nose? Because we've been trying to do that, and it's been really difficult.
Actually, what I did was I took the smallest dental brush I could get, and I used a shaver and shaved it down even further. So it's about a milliliter wide, and then was able to insert it with great, for me, difficulty. That's why I didn't use it. But that's actually a surgical approach, and I think it would be much easier in human, actually, right? And of course, we're not going to use polydocanol in human, but we'll be able to remove the infected tissue easily. But yeah, but practice, you can do it, I think.
Can you share which conditioning formulation you're looking at for FDA approval
You mean
instead of polydocanol?
we're not going to use polydocanol for human,
Right.
but we're going to use it in the mouse studies. Is that
Sure.
what you're saying?
Do you have a formulation that you are looking at using for human conditioning?
It's polydocanol.
For humans,
I'm
yeah.
sorry. For human is simply scraping the tissue to the bone that J.Car Nayak does in our lab routinely.
Do you have any other questions? Okay. If there are no other questions, let's thank our speaker.
All right, I'd like to welcome Alana Garrison, who's at Yale, working to advance nanoparticle delivery of CFTR mRNA. And I'll try to do the technical stuff here.
Top right. Exit.
Yeah. All right. I'll get your laser pointer. Should be good. Oh, sorry. Am I missing it? Finally.
Thank you. First, I'd like to thank the organizers for giving me the opportunity to present our work. I am Alana Garrison from the Egan Lab at Yale University, and I will be presenting on delivery of human CFTR mRNA using polymeric nanoparticles in a cystic fibrosis mouse model. So for disclosures, I have no relationships to disclose related to this presentation, but the PACE technology was made by Dr. Mark Saltzman and is licensed by Xanadu Bio. There are a couple additional author relationships to Xanadu Bio listed over here. So the background, which you guys have heard at the plenary and by other speakers, although highly effective modulator therapies are improving the health for many persons with CF, there are still many patients, the 6 to 12%, for which these therapies are not effective. For the patients who carry nonsense mutations, splicing, variants, or other mutations like large deletions, alternative approaches may be needed. And we believe that nucleic acid therapies may provide a way to restore CFTR for these patients. So we were looking at inhaled CFTR mRNA. There are several advantages, and one of the most exciting, especially for the CF Foundation, is that it's mutation agnostic. We want it to work for all patients regardless of their mutation. In addition, it can be targeted to the airway and delivered throughout the airways, whether you need it delivered to the trachea and bronchi or the small airways and alveoli. Our last major advantage is that mRNA only needs to be delivered to the cytosol to be translated into protein. It does not need to enter the nucleus. But, however, there are also many challenges when looking at inhaled mRNA delivery. One of the biggest challenges with any inhaled therapeutic, especially with CF lung disease, is that it can be trapped in the mucus and then cleared from the airway. Another challenge is making sure that the therapy is reaching the correct CFTR expressing cells, like secretory cells. Moreover, we want to know if there are any unforeseen off-target effects from delivering CFTR mRNA to cells that don't typically express CFTR. Lastly, we all know the effects of mRNA therapy are transient, so repeated dosing would be needed, but may not be tolerated or as effective over time. This may lead into another issue, that the particles may elicit an immune response, which could be detrimental, or it may be cleared by the host immune system, like being taken up by alveolar macrophage. Lastly, mRNA, especially human CFTR mRNA, is very large and fragile. So because the mRNA is so large and fragile, there are several strategies that can enhance its durability and stability. Just a fe' here. First, a 5' cap can be added, which can influence mRNA stability and translation. In addition, nucleotide modifications can be made, which can dampen the host's innate immune response. Also, changes in RNA secondary structure can be engineered into the mRNA that do not influence the protein sequence, but can influence translation rates. Lastly, like 3' and 5' UTRs can influence translation rates, stability, microRNA targeting, and localization. So specifically in our study, our mRNA is modified by adding a 5' cap that enhances stability, prevents degradation, and promotes translation by increasing the bonding affinity to initiation factors. We also have a base modification where our uridine is replaced with pseudouridine, which is known to dampen the host immune response by mirroring host mRNA modifications. And lastly, we also have a poly A tail that enhances stability. So in addition to modifying the mRNA also needs a delivery system that protects and effectively delivers the mRNA inside the targeted cells. So we delivered using PACE polymeric nanoparticles. PACE is also known as polyamine coester. It's a very versatile and tunable technology. It's a terpolymer, meaning it's made of 3 distinct monomers that can be arranged into different configurations to be formulated to many different types of delivery vehicles. It's biodegradable, and it has a mild cation charge, allowing for high mRNA loading and low toxicity. So specifically, our polyplexes have been optimized for airway delivery. We add a hydrophilic polyethylene glycol group, PEG, which can coat the surface of the airway delivery vehicle, which increases biocompatibility, decreases unwanted protein interactions, and also may reduce the toxicity of the polyplexes. So how it works is the PACE polymer associates with the mRNA through electrostatic interactions of the cationic polymer with the negatively charged phosphate groups in the mRNA, forming this polymeric nanoparticle that we often refer to as a polyplex that resembles a ball of yarn. So it's kind of like this. Instead of being in contrast to a phospholipid layer where lipid nanoparticles, where it's fully encapsulated, it kind of looks more like a ball of yarn. So our approach to determine if our nanoparticles loaded with human CFTR mRNA could restore CFTR function in our mice, we delivered the PACE loaded with human CFTR IN and IT, so in the nose and the trachea, to 2 different CF mouse models. Our first mouse model was a gut-corrected knockout model, but we have now moved on to testing in a model that contains 2 copies of the premature stop codon G542X, one of the mutations we hope that this therapy might provide a therapy for. So the GX animals are completely CFTR null and have no expressed CFTR. We express CFTR function and expression in vivo using the NPD assay, and ex vivo examining respiratory epithelia in oozing chambers to assess short circuit current. We also looked at inflammatory markers through the cytokines in the bowel and serum. Then we looked at CFTR expression in the tissues using immunofluorescence and Western blot. We did our evaluation at 24, 48, and 72-hour groups after delivery. In this presentation, we're showing our initial data from our GKO group. So, as some of the other presenters have said, we first started with we wanted to make sure that we could deliver to the respiratory tract. So we delivered PACE with a reporter mRNA sequence that encodes FLuc, which is a firefly luciferase enzyme. When the enzyme comes into its contact with its substrate, in this case D-luciferin, it emits light. The luminescence can be assessed at 24 hours after a treatment with IVUS imaging, which is an in vivo bioluminescence imaging system. So here are our results from that PACE FLuc delivery. As you can see in the middle, there is robust uptake and expression of the lungs of the treated animals that is absent in the control animals and in other organs, such as like the liver, heart, spleen, and kidneys. On the right, the bar graph quantifies the RLU, or the luminescence, per milligram of protein in the treated lungs versus the controls. These results reassured us we could effectively deliver PACE mRNA polyplexes to the respiratory tract, so we moved on to delivering PACE loaded with human CFTR mRNA instead. So when we looked at the mice after treatment, we looked to see if there was any evidence of functional CFTR respiration. We did this using NPD, which is a nasal potential difference assay. In this assay, the nasal epithelium is perfused with different solutions to assess function of ion transporters, including CFTR. So the CFTR is activated using a cyclic AMP stimulating cocktail, FSK, and IBMX. In the panel on the left, you can see 3 different NPD traces. The y-axis is in millivolts measured across the polarized nasal epithelium for each minute on the x-axis. As you can see in the black line, which represents a healthy wild-type non-CF animal, there's a 6 millivolt change or hyperpolarization in voltage when stimulated with FSK and IBMX, which is consistent with functional CFTR as we would expect to see in a wild-type animal. In contrast, this light blue line is a trace from a pre-treatment CF animal. It does not show this hyperpolarization due to the absence of CFTR. And then in contrast, the trace in blue is the post-treatment of the same exact mouse after treatment with PACE mRNA. And it demonstrates a significant hyperpolarization of negative 4.8 millivolts, suggesting partial restoration of CFTR function. So now if we move into the panel in the middle, the y-axis represents the change in NPD in response to forskolin and IBMX for each mouse that we treated, both pre-treatment and post-treatment. Each color-coded dot represents the same mouse before and after. As you can see, all of our mice exhibited an increased response to the cyclic AMP stimulating cocktail after treatment. We get this nice hyperpolarization, this nice downward slant. And then the last 2 panels show the PACE mRNA treated versus the control group. With treatment, there is a significant change in response, which is absent in the control, which stays about the same. So next, we wanted to see if we could identify CFTR in the lung tissue after treatment. After in vivo evaluation, our animals were sacrificed and lung tissue was fixed in OCT, then stained for DAPI and human CFTR. This is a zoomed-in scale of a larger slice of lung. We could see some alveoli here. On the panel on the right, there's detectable CFTR in the lung tissue that is absent on the untreated control on the left. So we are also able to identify mature CFTR using Western blot, this nice band C here, in the lung tissue at 24 and 72 hours. And again, it was not seen in the controls. And then the upper panel shows the relative expression of CFTR mRNA by RT-qPCR compared to a housekeeper gene, beta-actin. The treated is in blue and the controls are in gray. Clearly, you could see there is high expression in the treated tissue compared to the controls. In the next panel, it's the relative expression of human CFTR mRNA in nasal tissue, which again shows a marked increase compared to the controls. And lastly, it's the bowel pellet, so all the cells from the lavage of the lung, and it shows a very high level of CFTR mRNA expression, which could reflect uptake by alveolar macrophage or any remaining polyplex. So lastly, one of the most important things is we wanted to examine the immune response. So bowel fluid and serum were collected from the mice and analyzed for any evidence of an inflammatory response to the PACE mRNA. In total, 19 cytokines were assayed, but we only show a subset of the bowel results here. As you can see, there was no significant increase in measured cytokines through one-way ANOVA for any of the cytokines in the bowel here or the ones not shown, including and none in the serum either. In general, our histology and cell counts were unremarkable, so we did not show our data. But we will have additional cytokine data on the poster. It's number 276. It'll be at the end too. So in summary, PACE technology is an effective delivery vehicle of CFTR mRNA to the respiratory tract of CF mice. Just a single application results in partial restoration of CFTR function. Western blots and immunofluorescence confirm the presence of CFTR. So far, the single treatment induces a minimal immune response, but further evaluation of repeat dosing and immune response is ongoing. So looking towards the future, we are currently doing repeat dosing, where we're delivering every 72 hours and assessing every 48 to see if we have sustained CFTR expression, of course, investigating the immune response, and if there's any additional toxicity. And as I mentioned at the start, we are interested in the cell populations that are expressing and taking up the mRNA, so we plan to examine those as well. So for my acknowledgments, I would like to thank our whole lab group and everyone who supported me, the Egan and Bruce Shea labs on our floor, Dr. Suberi for starting the work, Dr. Lee for getting it to the finish line, James for running all of our wonderful Westerns, and Kelly for doing some cytokine data, and wonderful mentors, and especially my mentor, Dr. Egan, who without without her, I wouldn't know so much about CF. Like, I wouldn't even have the opportunity to be in CF research. And it's something I'm so passionate about. I have a family member with CF research, and I cannot thank her enough for all her wonderful experience and her patience with me and her teaching me and everything that she has done for me. I appreciate her so much. And of course, our funders, who without them, we wouldn't be able to do our research. And again, it is my poster is number 276, and the poster session is in a few hours, so I'll be there if you guys have any additional questions or want to look at that cytokine data. So thank you. Questions?
That was a really wonderful presentation. Are there any questions online?
Not online.
That was a really impressive talk. I'm curious about how about the persistent expression. How long do you think that you'll be able to re-administer before? Do you anticipate that that you'll be able to do every 72 hours long term, or is there anything that you think will develop over time?
So great question. That's something that we're definitely interested in investigating. So far we have seen like around 72 hours in our first dosing. That's when the functional data starts to decrease. So we want to keep it functional over time. So that's why we started with these animals. But so far with just our very small N, a repeated dosing, it looks like there isn't a very high immune response, but it's definitely something we have to investigate. But it also looks like the CFTR function is holding.
That's very exciting. Hillary, do you have a question?
Yeah. Hi, Hillary Valley from the CFFT lab. Very nice talk. Thank you. My question for you was, in one of your early slides, you showed kind of the importance of the transcript, the importance of their poly A tail and the promoter and your modifications in the sequence. And so I was wondering if you did any evaluation or optimization of your cargo. And when you show your partial functional rescue, do you think that is due to perhaps less than perfect delivery, or do you think that optimizing your cargo could help get you there as well?
So great question. I think, of course, optimizing our cargo could help us get it there. Unfortunately, I'm more on the, again, delivery/functional side than the design side. That's the Saltzman lab, who we think we have a great collaboration with. I'm not exactly sure if we did for this specific mRNA, what we did to look at the modifications, but I think they have done previous studies with other mRNA, and that's what we base this mRNA design on.
Thanks.
Hi, everybody. Anna Borrelli from TGEM Institute, Italy, and impressive results. And I was wondering if you have an idea of what kind of cells receive the mRNA.
So no, we don't, but that's what we're trying. We're trying to figure out a way to look at, possibly by tagging the cells to see where, because we're delivering it throughout the airway. So we want to know what cells are expressing it, and if those are like the functional cells that we think, like secretory cells, or if it's being picked up by cells that may not typically express it, what's happening. So it's definitely something that we're interested in. Great question. We're going to do that. So
thank you.
To be continued.
Okay, nothing else on there.
Hi, really great talk. Rana Ghosh, UT Austin. I was wondering, in the different paste compositions, do you have a sense of how much mRNA is being released into from the early endosomes?
So
degraded.
For this, we only use this one paste formulation. We're not changing the paste formulation off the top of my head. I am sorry, I'm not super familiar with the delivery vehicle.
Okay.
But there has been previous work on different formulations and which was best for airway delivery specifically, because that's what we want to look at. They did with COVID vaccination, actually, is what they were looking at. So there is work from the Saltzman group, but off the top of my head, I don't know. Thank you.
Short answer is we don't know.
Yeah, thanks, Alex.
If there are no more questions, let's thank our speaker.
All right, I'm excited to welcome Joe Porter, who is at Rochester working with John Look to advance suppressor tRNAs to address some of those premature stops that are so critical for cystic fibrosis. And yeah, welcome, Joe.
All right. I want to thank the organizers for inviting me to talk a little bit today about work in the Luick lab. encoding nonsense suppressor tRNAs in small DNA vectors.
Sorry. Are you good? I forgot. Yeah. So this will do the laser pointer. And that should be good. And then you can advance with these.
Okay.
With these.
Thank you.
Yeah.
Yep, there we go. All right, and disclosures related to this work. And so if we think about normal translation of proteins, basically that we get, if translation is proceeding normally, we have normal full-length functional protein. However, if there is a mutation that converts one of a coding codon to one of the 3 stop codons, we call this a nonsense mutation, and this results in either truncated non-functional protein or degradation of the mRNA transcript due to NMD. And so I think this audience can definitely appreciate the positive impact of correctors and potentiators for people with CF. However, for so-called class 1 mutations, which include PTCs, there's very little or no protein made. And so approaches to correct this will need to address mRNA or CFTR transcription or translation. And so the approach that we've been taking in the Lewick lab is so-called anticodon edited or ACE tRNAs, which is basically a type of nonsense suppressor tRNA. So normally in translation, tRNA is charged with its cognate amino acid by its cognate aminoacyl tRNA synthetase, which then this charged tRNA goes to recognize its cognate codon on the ribosome. So the idea with ACE tRNAs is that we change the anticodon so that it's now cognate to the PTC, and that this then seamlessly rescues the PTC in the ribosome. So if we think about PTCs in the context of CF, the most common ones are G542X, R553X, R1162X, and W1282X. And so work from Hillary Valley and the CFFT have produced 60HB cell lines that have been gene edited to contain these endogenous mutations genomically. And they've graciously provided these cell lines, which have been critical for this work. And I also want to point out that we use ACE tRNA leucine TGA suppressor for rescuing W1282X because it's been shown in work in David Bedwell's group that the W1282L CFTR has near wild-type function, and our leucine TGA suppressor tRNA has much higher function than the tryptophan TGA suppressor. And then I also just want to point out that Wuri Ko, a graduate student in the lab who's done a lot of the work that I'm going to show today, she has a poster where she's talking about extending this type of approach, basically using the leucine TGA suppressor for all of the most common CF mutations. So if you want to hear more about that, you can see our poster later. And so using these cell lines, we were able to show in a study published last year that ACE tRNAs can rescue both CFTR full-length, or rescue the CFTR mRNA transcript as well as the full-length protein. And in doing this study, we wanted to address, basically have a way that we could deliver, look at ACE tRNA rescue without having to correct for a deficiency of delivery. So basically the way that we addressed this was we just tagged an MD on green into the plasmid that's expressing ACE tRNAs. And so you can see in these typical flow sort experiments, basically we just have cell count on the y-axis, GFP or green fluorescence on the x-axis, and you can see if we transfect with an empty plasmid, we don't have green cells. Whereas if we transfect with a plasmid that's tagged with MD on green, we can show that we have about 50% delivery of the ACE tRNA by transfection. And so then whenever we went ahead with a variety of plasmids that we've tagged with MD on green that are expressing our ACE tRNAs. And whenever we're resorted for green cells and then isolated the RNA and then used RT-qPCR to quantify the amount of CFTR mRNA, she saw a rescue of anywhere between 41 to around 90% of rescue for G542X, R1162X, and W282X. And then when Matt Sipple, a graduate student in the lab, performed patch clamp electrophysiology, basically he patched on green cells, and he saw for R1162X about 75% rescue of the CFTR channel function and about 90% for W282X. And so what I hope this has kind of impressed upon you is that if we can select for efficient delivery of ACE tRNAs, that we can rescue near wild-type levels of the CFTR. But of course, that's an important caveat of being able to have efficient delivery. And so ACE tRNAs are still a genetic therapy, and that's a significant hurdle. But I think that ACE tRNAs have a number of positive attributes when it comes to being used as a therapeutic, a genetic therapy. Probably the first, most obvious one is how small tRNA genes are. So the entire gene, including the promoter and the transcription terminator, fits in about 150 base pairs. And then we can also deliver ACE tRNAs in a variety of formats. And those include as RNA, as DNA, whether that's naked DNA or as nanoparticle formulations. We can also use therapeutic viral vectors, including adenovirus, AAV, and lentivirus. But for today, I'm just going to be discussing DNA vectors. And so we think of sort of a typical DNA vector like a puck plasmid. This is good for an initial study where we're just trying to make enough ACE tRNA to study, but these vectors also have a number of drawbacks. So clearly the ACE tRNA makes up a small fraction of the vector pi, and also so that the other components cause excess bulk for the vector, which could have issues with intercellular trafficking. And also these bacterial sequences can cause silencing of the tRNA vector. So we wanted to look at some minimal DNA vector approaches that have been developed before. And so these have been developed in Mark Kay's lab as well as other labs. And so in this case, we're looking at both circular covalently closed as well as linear covalently closed vectors. And in the case of the linear ones, basically this pink circle just indicates that this 5' end is linked to its adjacent 3' end, and that's how it's covalently closed. And so in collaboration with Emily and Zach, undergraduates in the lab, we've been able to produce milligram quantities of these various picovectors. So in both cases, the substrate is basically just made by PCR of DNA. In the case of circular picovectors, these are made in a T4 DNA ligase-dependent reaction. Whereas for the linear picovectors, we use this pro-telomerase TEL-N, which recognizes TEL-RL sites that produce these covalently closed ends. And so these agarose gels is basically just showing that while the substrate is susceptible to T5X nucleus digestion, the covalently closed products are resistant, unless we linearize them with a BSU 361 restriction enzyme, and then they again become susceptible to T5X nucleus. So with these picovectors in hand, we wanted to just get a first pass idea of how they would function in cells. And so for this experiment, we just co-transfected the picovectors along with a plasmid that encodes nanoluciferase with TGA PTC and a wild-type firefly luciferase that serves as a transfection control. These were transfected with lipofectamine 2000 into HEK 293 T cells, and we assayed for luciferase activity using Promega dual luciferase kit. And so the normalized suppression ratio just represents for every well, we look at the nanoluciferase ratio to the firefly luciferase ratio, and then we then ratio that to a background where we have no ACE tRNA provided, and we use the nanoluciferase from expression from that, basically. And so in this case, we were looking all at leucine TGA suppressor picovectors, and as well as a scrambled negative control picovectors. And we looked at several different sizes. So we looked at 200 base pairs, 400 base pairs, 800 with 1 copy of the tRNA, and 800 with 4 copies of the tRNA. For both circular and linear, and then comparing with a 4x copy in a plasmid. So if we look at basically 400 base pairs and above for all picovectors, they showed robust PTC rescue. However, if we look at the 200 base pair circular picovector versus the 200 base pair linear picovector, you can see that we're not recovering function for the 200 base pair circular picovector. And this could possibly be because of the 2 different DNA structures may prevent different topological constraints. And so possibly this tRNA is just not being transcribed and we're not seeing function. One sort of fun anecdote from this is that we believe, based on what we know, based on our knowledge, that the 200 base pair linear picovector is the smallest functional expression vector that's been used. And one other thing to keep in mind is that there does seem to be some size dependence for rescue, but I think that could potentially be a bit of an artifact of the delivery method. I'll show a little bit more of that in a second. So we also want to look at how efficiently we can deliver these picovectors. And so we used a piggyback transposon that contains amnion green with a TGA stop. And so when Tyler Couch, a postdoc in the lab, did flow experiments, we could see that when we deliver our tRNAs, we rescue amnion green expression. And then this is just comparing with the read-through agent G418. And so we went ahead and did this with the picovectors. And again, Tyler saw that we're rescuing amnion green expression. And then whenever we isolated mRNA from the cells and looked at CFTR rescue, we could see significantly elevated rescue for the picovectors. And there you could kind of see that it looks like, in this case, we're electroporating the picovectors. And so I think that we see sort of less of a size dependence in this case. And then one other thing I should point out here is that we're actually not selecting for green cells. This is just from the entire population for the mRNA rescue. So we also did this with circular picovectors as well. So 800 scrambled parent plasmid, 1x800 and 4x800. We can see that we're getting about 80% efficiency of delivery in this case. And again, we're rescuing near wild type levels of CFTR mRNA, and this is without even selecting for the cells that had it delivered. We also wanted to look at protein rescue. So in this case, we were using a CFTR construct that has an N-terminal or a C-terminal nanoluciferase and either R1162X or W1282X PTC. And this allows us to do in-gel luminescence, so we can check basically for rescue of full-length CFTR. And so we saw both for R1162X, W1282X that we could rescue with our circular picovectors. And this basically kind of tracks what we saw with the mRNA, which is generally what we see is that mRNA rescue kind of tracks with the protein rescue. All right, so just a summary of what I've talked about today. So hopefully what I've showed is that with effective delivery, tRNAs can rescue near wild type levels of endogenous PTC containing mRNA and protein, and that we can encode these as small DNA picovectors, both circular and linear, and that these picovectors can meet or exceed the level of rescue for other vectors that we've used before. So I think there's some outstanding questions that we're still looking to address. And so one question is that based on the literature, we would expect that 200 base pair picovectors are small enough they should be able to diffuse into the nucleus in non-dividing cells. Obviously, this is important because we're delivering these tRNAs as DNA. They have to be transcribed to be functional. So this is something to keep in mind. And the other, I think, other point is that for other minimal vector systems, people have seen expression out to 6 months. And the question is just, we would expect that they would be the same for tRNAs, but this is something that we're still looking at now. And then just sort of the outlook of this, because I've talked a lot about different sizes, different cargos, but I think where we're really going with this is towards basically the 200 base pair linear picovector with leucine TGA suppressor, for a number of reasons. So I've sort of glossed over the production of these things, but it's not trivial. And I think that now that we've worked out a lot of the ins and outs of these production methods, that the producing linear picovectors is the most straightforward method. And so another thing that I didn't really get to talk about today, but we've been putting a lot of effort into optimizing the function of these tRNAs as expressed from DNA. And so we now have an tRNA leucine TGA suppressor DNA sequence that requires about 5 to 10-fold less DNA to rescue the same amount of a PTC. And then as well as I mentioned, WRI has shown that you can use the leucine TGA suppressor to rescue the top 4 most common CFPTCs. So I think all of this kind of points to us looking to use or move forward with this picovector as we're trying to move towards a therapeutic. And so for that, I'd like to thank John, particularly the whole LUIC lab, but I'd like to particularly thank John, WRI, Zach, and Emily, as well as our sources of funding. And with that, I'd be happy to take any questions.
All right, I can get us started with some submitted questions. One of them is a classic. It starts off really cool approach. I agree. The question is, they're wondering if you have a sense of the impact of disrupting stop codons broadly in a cell. So in other words, would you accumulate non-CFTR proteins with inappropriately translated products?
Yeah. Of course, if we're looking at rescuing PTCs, all transcripts are also terminated by what we call NTCs, natural termination codons. And so you would expect that these tRNAs will also read through those. So this was work that Jay did in the Sketch lab at the CFFT in collaboration with John. And so he did ribosomal profiling to actually look at where ribosomes are on transcripts. And so you would expect if we're reading through NTCs, the ribosomes would be in the 3' UTR. Basically. And so this is kind of showing that. And I'd say the key takeaways here, as it pertains to that question, is at most you see 2-fold increase in read-through of NTCs, whereas if you think back to what I was showing, we were seeing 1,500-fold rescue of the PTCs. So it definitely seems that tRNAs are much better at reading through PTCs and NTCs, but I don't think we've fully worked out why that is yet.
Hi, Robert from Tulane University. Great talk. It's really, really interesting work. I was wondering, especially like with this read-through problem, have you looked at any of if there's any innate nucleic acid sensing within cells due to kind of just different nucleic acids you've got in there?
So in terms of
does it activate like TLR sensors, RIG-I, and any of the cytosolic nucleic acid cell intrinsic inflammatory pathways?
Basically, if we deliver these vectors, are we activating these? Yeah, it's not something we've looked at yet, but we are going to for sure. Thanks.
Okay, we've got 2 more submitted questions that we'll go through. One question asks if you would expect any different risk of immune response when delivering circular versus linear picovectors.
That's a good question, and I've never considered this, and I'm not even sure. Yeah, I'm not sure how I would. Yeah, that's outside my area of expertise.
Yeah, I think there's some differences in when either of those is in the cytosol, the cell feels it differently. I think it's a good consideration. Another submitted question: what sort of delivery vehicles do you think would be compatible with this approach, and would there be a way to integrate them for permanent expression?
Yeah, that's a good question as well. And we haven't really, for myself at least, we haven't really gotten there. I would say in terms of, I think we're in a bit of an interesting space, I think, because most DNA vectors are never this small, right? And so I think we potentially have access to delivery vehicles that maybe would be more RNA-like potentially, because we don't have to, if we can actually, if it diffuses into the nucleus, we maybe could use some different types of delivery approaches that have been used before for DNA. But there again, I don't know, because we haven't quite got to that yet. What was it there?
I think you covered it.
Okay, sounds good.
Would you expect to see any immune response? I mean, not to the particles themselves, but to the extensions in protein. I guess this applies to gene therapy as well, but because you have potential of extending a whole lot of additional proteins, how would you sort of deal with that?
Yeah, that's definitely a concern for sure. And I think there's kind of been, this is something that people have looked at, especially even for total gene replacement for sure. And I think there's sort of conflicting evidence of how much of an immune response is seen from this, particularly, well, there's kind of two parts to that answer. If you're just looking at an immune response to now having the full length wild type protein made, I think generally that even for these ones where you have very small amounts of the protein made, the immune system has kind of already seen that protein, so it's usually pretty much okay. Now, in terms of the, there would be C-terminal extensions reading through NTCs. I think there again, you get the fidelity of translation is only 1 in 10,000 anyway. So likely there is some amount of these C-terminal extensions that are existing anyway. So there again, I don't think people have looked at that closely yet, but this is definitely something to consider.
We're doing quite well on time, so I'm going to ask a question.
Okay.
Have you, in your design of your vectors, have you looked at incorporating a nuclear target? And do you think you'd see increased efficiency if everything that you delivered got straight to the nucleus?
We've been considering this. We haven't done it yet, but yeah, likely that would probably, yes, I think that would help.
Hi, it's a great presentation and great work. Did you consider the look at the non-stop mRNA decay as behind the global mRNA decrease?
Mm-hmm. We haven't specifically looked at that, no. But
you cannot screen for the ribosomal profiling for that, because that's never made to a protein.
Yeah.
Okay. If there are no more questions, thank you for a very lively discussion.
Thanks, everyone.
And enjoy the rest of the meeting.